Um, Thank you, Jenny, for recording. Um, if you have not done so already, first of all, welcome and thank you all so much for your time and for volunteering this season with us, and for attending the live session of the Zoom training. Um, if you have not done so already, please make sure that you are muted and that your video is shut off until we reach a Q&A portion at the end of this presentation. Um, my name is Eileen Fitzgerald, she, her, and I'm the Education and Outreach Associate with the Lake Champlain Committee. And I'm also the coordinator for the Champlain Aquatic Invasive Monitoring Program, or CHAMP. And I'm joined today by our project officer, Meg Modley of the Lake Champlain Basin Program. She is the Healthy Ecosystems and AIS Management Coordinator of the Basin Program. And I'm also joined by my colleagues, Dr. Lindsey Carlson, who is LCC's Water and Science Coordinator and also the manager for CHAMP, and as well as Jenny Patterson, who is the Executive Director of the Lake Champlain Committee. So I'll just pass it quickly to Jenny to say hello to everybody. Jenny, go ahead.
Thank you, Eileen, and hello, everybody. As Eileen said, I'm Jenny Patterson. I'm the Executive Director of the Lake Champlain Committee. I started in that role in January of 2025, following Lori Fisher's retirement. And you may well know Lori. She led LCC for many years and developed the cyanobacteria monitoring program and also initiated this CHAMP program. So I'm super excited to be here on this training because I was a CHAMP volunteer myself last year. I loved it. I learned so much. I especially appreciated the in-person training as an opportunity to get to meet Eileen and other CHAMP monitors in person and to actually get hands-on with the materials. So my one word of advice is please pay close attention to the instructions on how to throw the rake because I clearly did not pay enough attention myself. And the first time I threw it, I had to wade back into the water and get it. But I did learn from that experience. Without further ado, I'm going to turn the training back over to Eileen and Meg, and I'll stay on. We'll have some question and answer periods, and I love to hear people's questions because I learned so much. So I will be there for that. And thanks to all of you for volunteering. Thanks to the Lake Champlain Basin Program and NUIPIC for supporting this work that LCC is doing. And let's get on with the training.
Thank you, Jenny. Just a couple quick housekeeping things before we really get into it. This is a Zoom training and we're recording it so we can share it with the volunteers who can't be attending right now. So we ask that people remain muted with the video off until the end of the training. To mute your microphone, you can do this on the bottom panel of Zoom. Click the microphone icon and it should have a red slash through it. And same goes with video. And then as we go through, I encourage folks to use the chat feature to ask questions. You just click chat and then it'll do this pop-up on the side, drop your question in there, and I'll pause at one point throughout the presentation before I go into the survey methods to go over some questions, and then we'll do a larger Q&A session at the end of the training. So what this presentation will cover is first a broad overview of aquatic invasive species, also referred to as AIS, and why they're an issue in Lake Champlain, and what sort of everyday folks can do to prevent the introduction and the spread of aquatic invasive species. And then I'm going to move to what is this program, the Champlain Aquatic Invasive Monitoring Program, just a little bit of its history, how it works, what we're asking of volunteers, what we provide to you as organizations supporting you as volunteers. And then we'll go through what are the aquatic invasive species we need to look out for in Lake Champlain, and then a step-by-step on how to conduct and submit a survey for CHAMP. With that, I'll pass it over to Meg to do so we can learn about aquatic invasive species.
Thanks, Eileen, and good afternoon, everyone. Again, my name's Meg Modley. I'm with the Lake Champlain Basin Program, and I'm super energized coming off of our first weekend of Memorial Day weekend of being out on the boat launches, inspecting watercraft and trailer, and today a very zebra mussel encrusted dock that was going to float out into Lake Champlain. We made sure there weren't quagga mussels on there. Um, so welcome to the training, and I will just start off with the basics, sort of the one-on-one of what aquatic invasive species are. So when we talk about aquatic invasive species, we have a very specific definition. We are talking about plants, animals, and pathogens. So all taxa really that are not native to this area or Lake Champlain pre-European settlement, but also cause some kind of harm. And that can be to the economy, to the environment, or to human health. And it takes a while for non-native species, once they're introduced into the lake, to cause impacts that we are able to measure. So right now, one of the species that's sort of on the fence would be a fish called the tench, which came into the northern end of the lake, and we started seeing it here and there. But now the state agencies are finding them everywhere, and they're increasing in size and number, and it's quite likely that they're displacing some of our other native species. So they're teetering on that edge of being able to cause some harm that we're able to measure. But the three prevalent invasive species that you will see or might be familiar with in Lake Champlain include submersed Eurasian water milfoil. On the left-hand photograph, you'll see a really dense bed of Eurasian water milfoil. The center photograph is a combination of fishhook and spiny water flea, and you're looking at thousands of them accumulated on a downrigger that's coming out of the lake. And these organisms are little crustaceans that don't seek out the line, but get caught or hooked up on the line as you move it through the water. And then they foul the line, whether it's a fishing line or a downrigger. And then on the right, our very famous striped zebra mussel that is famous for cutting the feet of swimmers, encrusting our historic shipwrecks, and fouling a lot of the infrastructure along the shorelines of Lake Champlain, as well as water intake pipes. Next slide. So there's a number of different ways by which these organisms can move across the landscape, and most of them are with assistance from us. Um, so there is often unintentional or intentional bait fish introduction. We see in Lake Carmi, there was an introduction of alewife that was likely an intentional introduction, maybe by an angler that wanted to create some new source of food for their sport fish that they were interested in. But the bait fish introduction pathway is significant, and it was one that caused a lot of the emergency bait fish regulations to go into effect in 2007 and 2008. And that was primarily because not only were bait fish being moved illegally, the water that housed the bait fish could carry a pathogen called viral hemorrhagic septicemia, which we don't have in Lake Champlain yet and can impact over 25 different species of freshwater fish. Another pathway is most often unintentional, water guarding or aquarium release. These are, sorry, not aquarium release, but water garden ponds. Those are situations where people will plant non-native species and sometimes mislabeled invasive species in their backyard, and then we will have a tropical storm or a flooding event, and those species quickly are washed into a tributary and may go into the receiving waters of pick your favorite lake. So it's really important to pay attention to the native species that we're planting in our gardens and our landscapes, and making sure that those can't get out if they are not native. So aquarium dumping is intentional. It's usually for a good reason. People feel like they're trying to free Nemo or save their favorite animal that they can no longer keep in their aquarium or is outgrowing their space, so they dump it into a water body. But unfortunately, those can often lead to reproducing populations and new infestations of species in the lake. And then One that we have a huge education, outreach, and spread prevention program for is the overland transport of aquatic plants and animals. Um, and the animals is the important piece there. There are early life stages of many aquatic animals that are not visible with the naked eye, that can be transported in water, that could be in the motors or in the bilge or live well of watercraft. So the overland transport on boats and trailers is not just removing aquatic plants or macrophytes, but really making sure that the watercraft are drained and or dry to make sure that we're not moving the early life stages of these organisms around. And then finally, canalways. Lake Champlain is connected to the Great Lakes via the Champlain Canal to our south and the Erie Canal that then goes west. We're also connected to the St. Lawrence Seaway by the Chambly Canal to our north on the Richelieu River that takes us down to the St. Lawrence Seaway. Thanks, Eileen. So two important graphics that we have updated in our State of the Lake report for Lake Champlain in 2024 include tracking the number of known non-native and invasive species introductions in the left-hand chart. And what you're seeing is the number of these organisms and when we think they became introduced into Lake Champlain. I want you to note that the lighter green colored organisms are what we consider non-native and have not been measured to cause some kind of harm yet. And some of those are intentional introductions. You'll see rainbow trout. You'll see largemouth bass. These were non-native species that were introduced for our recreational use and enjoyment, but they are not native to Lake Champlain. Whereas the dark colored organisms, of which there are just over a dozen in Lake Champlain, include water chestnut, Eurasian water milfoil, the fishhook and spiny water flea, etc., the zebra mussel. These are organisms that we know or have measured to cause some kind of harm. So the last known established population that was detected was in 2018, and that was the fishhook water flea, and that was detected by our biological long-term water quality monitoring program on Lake Champlain. But just last fall, the CHAMP program volunteer itself discovered Corbicula fluminea, which is golden clam or basket clam, in the southern end of Lake Champlain. And it's very likely that that will be determined to be an established population once the state agencies do some more research and investigation. And then on the right-hand side graphic, this is pretty popular and effective in expressing the amount of pressure that we have in Lake Champlain of known non-native and invasive species, but also that we are connected to other water bodies like the Great Lakes, the St. Lawrence, and the Hudson River that have a greater number of known non-native invasive species. So Lake Champlain, we have a lot to protect. We have we don't have a lot of organisms present in those other water bodies, but we're also a source lake for uninfested water bodies in the Adirondacks and in the state of Vermont. So we're particularly concerned with watercraft and organisms possibly exiting Lake Champlain and transiting to an uninfested water body in our region. Next slide. So why are they an issue when they get into Lake Champlain? Well, many of you probably have your own personal stories of your recreational experiences, whether that's boating or swimming or sunset cruising or fishing in the lake, and how those experiences have been impacted by invasive species. But most of these organisms are really great ecosystem engineers. They come into a body of water or a disturbed area, and they're able to monopolize on the different resources that are available. So most of these plants and animals, when they are introduced, lack the predators that they have in their places of origin, whether those are in Eurasia or the Ponto-Caspian Sea or China or some other location. The predators just aren't here yet and haven't figured out how to take advantage of these new organisms. And they also come into a system that is relatively free of diseases and parasites that they're used to in their place of origin. They tend to reproduce very frequently and in varied conditions. So the pH levels can be varied, the salinity can be varied. they're really tolerant of these different conditions. So they're able to reproduce early, often, and again, monopolize those resources. So that's the main points of how they become an issue in Lake Champlain is they get in, they reproduce, and they have few predators in this new environment. And as you can see, this is a really dense bed of water chestnut in the southern end of the lake, but another example again of how these organisms can monopolize new environments. And here, water chestnut is topped out at the water's surface. It looks more like a putting green. There are reduced oxygen levels underneath these dense mats of water chestnut where it's very unlikely that fish can utilize the habitat or other organisms can live under these dense mats in the southern end of the lake. Some of the invasive species it's okay. I mean, some of the invasive species can carry other types of parasites or diseases that then become an issue for our native species, or they might carry toxins. Some plants can cause, burns on the skin. Some organisms can carry parasites that might be dangerous to other organisms or humans themselves. So there's a number of different ways these invasives can also impact other organisms. So what can we do about it? We have a lot of education and outreach and spread prevention programs. There are some signature outreach programs called Don't Let It Loose. That's primarily for pet and aquarium owners and education about how to properly house your pets if they become too big, or proper euthanasia protocols for those organisms so they don't come into a new environment and destroy native populations. Clean, Drain, Dry is another national aquatic invasive species spread prevention message, much like Stop Aquatic Hitchhikers, and talks about the importance of people taking best management practices by removing what they see, by draining water, and by drying their recreational equipment and their trailers and their watercraft in particular to prevent the spread of invasive species. And the big thing that we like to remind folks is that it's really important to target your messages to different users on the lake. So we have different types of outreach materials when we're talking to children versus adults, or an angler versus somebody who's going out to swim. And then we also want to be aware that we want to have some of these educational and outreach materials translated in different languages. We have a lot of French Canadians who utilize Lake Champlain, but we have a number of new American communities that live in and around our Plattsburgh and our Burlington areas, and making sure that they have access to this information and the messaging.
Okay, thanks, Meg. So just to give an overview of another thing to do about the harm that aquatic invasive species can have on Lake Champlain, which is where you guys come in. I just want to give an overview of the program. So CHAMP is a community science program run by the Lake Champlain Committee, or LCC. We're a bi-state watershed-based nonprofit dedicated to using science-based advocacy, education, and collaborative action to safeguard habitat, promote access, restore water quality, and foster stewardship in the Lake Champlain watershed. A major component of our work is community science, which is a form of scientific research that provides everyone, regardless of background, an opportunity to contribute meaningfully to the data on, in this case, aquatic invasive species. LCC manages a similar program that has been around for a lot longer, which is the cyanobacteria monitoring program. A lot of you folks are also in that. So community science allows you to be the eyes and the ears on the ground for us, and also for state agencies who manage aquatic invasive species, and for researchers trying to better understand the spread of aquatic invasives. So CHAMP started in 2023 in response to state agency need to survey for aquatic invasive species on Lake Champlain. Vermont and the Adirondacks both have really robust volunteer programs to monitor for AIS in lakes throughout those regions, but there's just been trouble getting coverage on Lake Champlain, so that's sort of where we stepped in. And we stepped in with support from the Lake Champlain Basin Program as well as New EPIC, and we piloted CHAMP in 2023 and grew it further last year, where we covered 19 sites, and we're hoping to do a lot more this year. But through CHAMP, we train volunteers to identify aquatic invasive species, and we train them in survey methods. And then throughout the season, we empower them to conduct surveys and report their findings back to us. These findings inform research, management, and very importantly, early detection, rapid response actions. Because for invasive species, being able to act early makes a massive difference in controlling populations and spreads. So through this program, you'll learn the 8 target aquatic invasive species that have already been detected in Lake Champlain, as well as 8 watch list species that have been detected that have not been detected in Lake Champlain, but have been found in the Hudson River, in the Great Lakes, in the St. Lawrence. I think back to that map that Meg showed, how we're hydrologically connected to these water bodies. We're just training we want to train you in identifying some species that could be potential invaders. And like I said, you'll also learn how to conduct and submit those surveys. So as a CHAMP volunteer, you will participate in or watch this virtual training session. And you'll also attend an in-person training session in early mid-June. You should have already registered for this on your sign-up form, but please let me know if you have questions on that. And then you will conduct and submit at least 3 surveys for these species that we train you in at your site between July and October. Ignore the fact that that says boat launch site. That's from a previous training. I'll go over site specifications. They don't have to be at a boat launch. But yeah, your trainings or your surveys will take place between July and October. And then we will support you with these trainings, as well as a toolkit, which includes a very comprehensive manual that has identification resources, survey instructions, survey data sheets, reporting guidance, contact information, loops, which are little magnifying glasses that you can use to help you identify smaller plants and mussels, survey rakes, and a bin for observing species. And we'll also be there for ongoing technical support throughout the field season. So what do we survey for in this specifically? We have 8 species that are established in Lake Champlain. That includes brittle naiad, curly leaf pondweed, Eurasian water milfoil, European frogbit, golden clam, which is not necessarily established, but it has been found in Lake Champlain. So we moved it to the target species list this year. We also have water chestnut, variable leaf water milfoil, and zebra mussel. And then on the watch list, we have Brazilian waterweed, Carolina fanwort, hydrilla, parrot feather, starry stonewort, water soldier, round goby, and quagga mussel. And I'll pass it back to Meg to give an overview of each of these species.
Great. So we've got a few, so we'll go through them and hopefully you'll learn a thing or two, but you can always refer back to this recording if you need a refresher. So we're going to start with the species that are already present in Lake Champlain. These are just nice photographs of the 8 species that we will dive into. So let's get going. So the first one is brittle naiad. This is one that I think in Lake Champlain you will see not as frequently. You can go to the next slide. You will see that this is a really delicate plant that tends to grow on the sediment interface, and it grows only, a few inches up off the bottom of the water. So this doesn't grow to the surface of the water as Eurasian water milfoil or curly leaf pondweed do out in 15 to 20 feet of water. So it's a submerged species. It's native to South America. It has been confirmed in Lake Champlain and a small handful of other water bodies in Vermont and New York, but it is not as prevalent or widespread. So in our boat launch steward program, we do see it occasionally because watercraft come and go in those shallow areas, coming and going from the lake. And in some places, there is brittle naiad, but you're going to want to look for some key characteristics on brittle naiad on the next slide. So this plant, it can look at quick glance like many different species in Lake Champlain, and you really have to float it and put it on a white background to start to distinguish this one apart from other species. So the key pieces about brittle naiad, it has opposite leaves instead of alternate leaves. So they're opposite, and they look almost like little feathers, and they're serrated. So it although I guess the description says that it looks like it's whorled around the stem, meaning it has multiple leaves that come out from the same point on the stem. It's not a whorled arrangement, but an opposite leaf arrangement. So leaves come off from the stem opposite. Next slide. And it does look like some other natives. So we have a few native naiads in the lake, neither of which have this serrated leaf edge. So it's this one, I often even have to get out my identification guide and really compare and try and determine if I'm looking at a native naiad or an invasive naiad species. So just when you see opposite leaves with serrations on them, you may want to get out the guide and take a closer look at this organism. Okay, next one. The next one you can tell apart from all of our other pondweed species in Lake Champlain. So we have many potamogeton species in Lake Champlain, and all of them are native except for curly leaf pondweed. And curly leaf pondweed really does look like lasagna noodle leaves. I've found very few instances where it doesn't. So this one grows also in up to 15 to 20 feet of water. It will grow all the way to the surface of the water. Let's see, it's confirmed in many different bodies of water. I would say in some of them it is problematic, in others it is not. It just depends. I have seen really dense beds of it in the southern end of Lake Champlain and in a few other water bodies, but it is confirmed in many water bodies in the Northeast and surrounding Lake Champlain. Next slide. So here you can get up a little bit closer. The lasagna noodles do have that serrated leaf edge. So that little jagged edge that you see on the left side. They're really these long lance-shaped leaves, and they do curl. I wouldn't always look for the little flower bract that you see in the right-hand slide unless it's growing up to the water surface, because you're only going to find those really at the tip of the plant. This is a plant that's going to spread mostly by seed and turion. So if it fragments, that's not a concern. What the spread mechanism or the reproductive mechanism here is really seed and turion. Next slide. Eurasian water milfoil. This one you will see a lot. Myriophyllum spicatum has a few defining features, and when you get to see this one in the field, please pick it up and hold it and play around with it. You'll get a feel for what Eurasian water milfoil looks like. It is distinct from our other milfoils and other species, native species in the lake. Next slide. So we've seen already a lot of Eurasian water milfoil. It grows early and often in the field season. It is a submersed plant. It can have red tips at the end of the growth. Either if fish are eating on it, it could be a sign of some damage, or just the tip growing towards the surface of the water. It's not always red. I don't go by color. What you really want to go for is the structure of the plant here. So it's all over. Confirmed in many different lakes. And let's go to the next slide to get into the specifics. What you want to know about Eurasian water milfoil is that on each leaf, and there are 4 leaves on a whorl that come off of the stem of the plant, on each leaf, there will be more than 12 leaflets. So on that feather, more than 12 individual little strings coming off one side of the feather. Or more. It could be up to 18 or 19. that's likely Eurasian water milfoil. If it has less than that, it may be a native species. But we do have some hybridization. Another piece about Eurasian water milfoil, often the internodal space, which is the space on the stem between where the whorls come off the stem, is greater than on other native species and definitely on variable leaf. Those variable leaf milfoil, the whorls tend to be stacked almost on top of each other. And when you pull this one out of the water, it kind of droops along the stem. It doesn't have great structure. But Eurasian water milfoil is prevalent. I'm sure you've seen it and you will see more of it. And then when it reaches the surface of the water, it will extend up. It's bract, which has the flowers, which can be really brittle. And if you just see that part of the plant, you probably won't know what it is, but you have to take the pieces underneath the water. But this plant does spread by fragmentation. So it really only takes, a node of the plant to become fragmented and go and settle on the sediment bottom of the lake to reproduce again. All right, another one. So European frogbit is a floating plant. It's not rooted to the bottom of the lake. It grows out. It has these very distinct leaves. Let's go to the next slide. It does have a white flower, but it's unlikely that, it only happens for a week or two of the summer. So when you see European frogbit, what you're looking for is it floated or is it rooted? And if it's rooted, you may get a broken stem that comes up from the bottom of the lake, or it may be attached to a really big rhizome root. That's usually a native lily or some other kind of a water shield kind of plant. But these are floating, invasive, and they tend to grow in the shallower, calmer backwaters. It can get very dense in places. There are very active management programs with the Lewis Creek Association in Town Farm Bay, but other places are, it's just sort of growing and is a nuisance, mostly for ducks because they don't eat it. Let's go to the next slide to look at the structure of the plant a little bit better. So here you can see multiple plants can be connected together by these stolons. So if you pick up one, you may end up with a whole, train of them coming out of the water. But again, they're not rooted to the bottom. They have these heart-shaped leaves that are distinct from other pond lily leaves that either have sort of pointed tips to their lobes or have a more circular leaf at the surface. So again, we're going to want to look at the structure of the plant and note if it's rooted or not rooted, and look at that leaf outline so that you can figure out if that shape matches European frogbit. Okay, unfortunately moved on to the in Lake Champlain list, the golden clam or Corbicula fluminea. So Corbicula fluminea has only been found so far in the southern end of the lake at the South Lake Boat Launch in New York, but we do have 3 surrounding populations in Lake Bomazene, in the Champlain Canal, and in Lake George. And all 3 of those water bodies are hydrologically connected to Lake Champlain. The telltale about golden clam is that it has raised concentric ridges. And that really means that if you pick up the shell and you have any fingernail at all, you can click it across the shell. Our native fingernail clams do have these lines on them, but they're not raised. So it's more of a smooth shell. So all of our native clams tend to have smooth shells, and the golden clam is the only one we know of that has these raised concentric ridges on it. So these are hermaphrodites, which means it only takes one that has the reproductive organs in it to produce many, many more. They are from Asia. We believe they could have been introduced either in contaminated soil or at this point now overland on boats and trailers. The early life stage of these are similar to zebra mussels. They have small juveniles that float in the water column and settle in the sediments and then grow. But they do not have the byssal threads or the attachment of the zebra mussels. Zebra mussels have these little threads that they can glue onto things with, and The golden clam don't do that. They live in sandier substrates, which is different than the zebra mussels. They can vary in color. They can be yellow, they can be purple. So again, I'd look more at the structure of the shell, and note where you're finding them. They can grow in areas where they're plants, but they prefer sort of a coarser sandy sediment. All right. Yeah, this is again, these are ecosystem engineers. They can reproduce multiple times a year, and in some cases, I think more than 4 or 5 times a year. They can produce many offspring, and they can grow in really, really dense numbers. And they've been observed in some hundreds of thousands in square meters in Lake Tahoe. So it's interesting, but they are not doing that well in Lake Champlain yet. Okay. Variable leaf water milfoil. Again, it looks similar to Eurasian water milfoil. Let's look at the structure of the plant. We call it more of like a bottle brush appearance in the water when it's growing and healthy. It's again a submersed plant. It can spread by fragmentation. Um, and it was first found in Lake Champlain up near the Missisquoi National Wildlife Refuge, and then found in the southern end of the lake down by Crown Point. It's not widespread throughout Lake Champlain, but it's probably further spread than we're aware of because we don't have enough of you out there looking yet. So it's really helpful to have you guys out there and collecting samples so we can take a look at what you're finding. Um, it's confirmed in many other New England states. It's more prevalent in New York than in Vermont. And the Halls Lake infestation, oh, we were so close to eradicating that one, and I have to check on how it's doing. But let's go to the next slide for the number of, oh, they're lookalikes. Okay, so again, for variable leaf water milfoil, the number of leaves on or leaflets on the leaf can vary. And they tend to be clumped closer together on the stem. So they have a whorl with 4 leaves, and it's usually, again, greater than 12 leaflets on a leaf that comes off of the whorl. Okay. Native submerged lookalikes. Well, they any of these can look like Eurasian water milfoil, but when you take them out of the water and you float them with a white background, you'll see that there are differences. We have a lot of Utricularia species on the left-hand side. These are our bladderworts, and they're native. Primarily, bladderwort's a really cool aquatic plant. It has these little pockets that are on the leaves themselves. Those are the little dots on there. And those are little pouches that they eat with. So they turn brown or black when they're full, and they're translucent or light green when they're empty. So it's a plant that takes advantage of some of the nutrients and the organisms in the water by grabbing them in the water. So I think bladderwort's one of the cooler plants out there. The one in the right-hand side is our native um, coontail species. And coontail can look like Eurasian water milfoil, but when you look up closely at the leaves around the whorl on the black and white photo in the lower right-hand side, you'll see that they're forked leaves. They're not feathers. So these tend to be spiky and forked, and it is more of a brittle plant. So when you pull it out of the water, it has more structure around the stem. And it's a very common species. You will see coontail along the shores of Lake Champlain almost everywhere that I go. So I expect you will see a lot of coontail this year. Okay, water chestnut. This is a unique plant, a unique invasive plant in Lake Champlain. It has these triangular serrated leaves, and it has little bladders on the leaf stems that are air pockets that basically float the plant up to the surface. And that one is surrounded by a bunch of our native duckweed. Let's go to the next slide. So water chestnut is an annual plant. We are aggressively managing water chestnut in Lake Champlain with mechanical harvesting and hand harvesting. It was a water garden escape. It actually came out of one of the one of the botanical gardens in New York, escaped into the Hudson and then moved up through the Champlain Canal into the southern end of Lake Champlain. It's been managed for decades. We're now spending over $700,000 a year removing it from the lake. And the reason we do that is because it's an annual, it has a nutlet. So let's go to the next slide. And the nut is that in the top right-hand corner, that seed or nutlet, and when it's dry, it's black and it's spiky. It almost looks like an alien head. And it can puncture your foot or your hand. You need to be careful when you're harvesting. You can slice your fingers on them. But they're green in the picture below when they're wet or when they're alive. And these little nutlets, if you can remove them on the plant before they drop off into the sediment, you can remove the threat of them regrowing. The challenge is the nutlets can be viable in the sediment for quite a number of years. A very small percentage of them can be viable for over 10 years. But that's why we have to consistently go back and manage year after year to deplete the population. So that's the species that's in my background here, creating the mat behind me. So this one is very easy to tell where it is. Your primary concern here is removing those nutlets and getting them out. The roots can also possibly regrow a plant. So pay attention when we're harvesting it. We try and get the entire plant out by gently pulling on the plant stem to dislodge it from the sediment. But again, if we're getting those nutlets out of the water, that's the success. Note that the stem of the water chestnut could look like Eurasian water milfoil. You need to take it out of the water and look at the plant in its entirety to get a good idea of what you're looking at. Okay, next slide. All right, then we have the zebra mussel. Zebra mussels can come in many different sizes, different patterns, a little bit of different colors, white, brown, black. They are a nuisance. So this is a mussel that came from the Black and Caspian Seas. Like most of our invasives come in ballast water or fouling the bottom of international watercraft that are transiting the ocean, come into the Great Lakes and dump into the Great Lakes. They get fouled and then those organisms make their way, unfortunately, into Lake Champlain. So unfortunately, we have zebra mussel, but we don't have its evil twin, the quagga mussel, yet. So it's a small freshwater bivalve. It can filter 1 to 2 liters of water a day. That can have great impacts to the ecosystem in the lake by removing a lot of the small biota at the base of the food web and concentrate a number of different things, whether they're concerning chemicals or biota. They can concentrate them at that lake bed interface where they grow. They are the only mussel that we have in Lake Champlain that attaches to things. So our native mussels grow mostly in soft sediments and are doing just fine until zebra mussels come and grow on top of them. And when they do that, they can suffocate the native clams and make them unfit enough so that they can starve or die. Let's go to the next slide. So we do see these fouling boats and docks and many things. And again, it's the phenology or the life history of this organism that makes it so successful. These zebra mussels can lay eggs many times throughout the year as soon as the temperatures rise above 50 degrees, which it is almost ubiquitous across the lake today. It's almost above 50 degrees everywhere. So the eggs will hatch into juveniles that float and then settle back down and can regrow into mussels. So they spread mostly by the juveniles floating in water currents, but in the unfortunate cases where zebra mussels attach to the bottom of boats or motors and move to a new body of water, they can fall off and regrow in new bodies of water. They are huge biofouls. I know most of our water intake facilities need to take measures to clean out their systems of zebra mussels. I know our hatcheries have to remove them from their intakes. We're concerned about them encrusting our historic shipwrecks on the bottom of the lake and cutting the feet of swimmers. So those are the species that we have present in the lake that you will likely see, with the exception maybe of the brittle naiad. I think you'll see most of the species on that list, unless, except for golden clam, unless you're at the South Bay Boat Launch. But we're going to switch over to some of the species that we don't have in Lake Champlain yet, and that we, look similar to some of our natives, that we want you to keep an extra eye out for. So let's jump into these 8. All right. The first one is Brazilian waterweed, or Aguirrea densa. This species looks a lot like our native Elodea or waterweed. It has a distinguishing feature in that it has greater than 3 leaves around a whorl on the stem of the plant. So this is a submersed species. It's from South America. It's not yet found in Lake Champlain. And if you see something that has more than 3 simple leaves on the whorl, you're going to want to collect that and report that back pretty quickly. So let's go to the next slide with a little bit more detail. So here you can see the leaves are smooth, but there's more than 3. So these are lance-shaped leaves, and they come in whorls of 3 to 6. They can vary, but as soon as you see more than 3 leaves on the whorl consistently, this is something you're going to want to let us know about. I haven't ever seen Brazilian waterweed in our lake or in our region, so hopefully you won't either. Let's go to the next one. And very similarly looking, and the difference here with hydrilla is that it does have a serrated leaf edge. So let's go. So hydrilla verticillata is another aggressive aquatic weed that looks like our native Elodea. Again, not found in Lake Champlain, but present in the Connecticut River, in the Croton River in New York, in Cayuga Lake in New York. So there are a few infestations that are relatively close by that could be introduced. So it's not present in Lake Champlain yet. When we talk to the Army Corps of Engineers that does most of the research on aquatic plant management and control, they think that this plant is worse than Eurasian water milfoil, if it were to get into Lake Champlain. So here are some of the distinguishing features of hydrilla. Again, it has that serrated leaf edge, and if there are more than 3 leaves on the whorl, again, whether they're serrated or smooth, please report it, because it could be Brazilian or hydrilla. Most of the hydrilla plants in the southeast have tubers, and the tubers in the lower right-hand photograph, that white fleshy material, is the sort of the root ball that grows in the sediment, and the tubers can lay dormant over a winter season and regrow. But there's a new form of hydrilla in the Connecticut River that doesn't have tubers. So we're not going to call that a defining characteristic, but if you find a white fleshy tuber at the base of the plant root and there's more than 3 leaves on the whorl, that one we're going to pay attention to. This one's spreading not by fragmentation, but more by seeds and turions. So you see the turions grow in the place where the leaves come into contact with the stem. And then there's some other seeds that can grow off of the hydrilla, and that's how they spread primarily through currents. But waterfowl do like to eat them. They are, again, submersed. They're not going to come out of the water, but they can grow up to the surface of the water. Okay, so here's some of the lookalikes. So again, the Brazilian and the hydrilla are more than 3 leaves on the whorl. Whether they're smooth or serrated, it's a problem. And then we have our common waterweed, which is almost all the time 3 leaves on the whorl, smooth. Slender waterweed does also occur in Lake Champlain. It also strictly has 3 leaves on the whorl. Slender waterweed can hybridize with our common waterweed, and it looks fishy. And we've encountered that, and we've sent it away for genetic analysis and just found it's a native and a native that are hybridizing, and not of concern, but good to know that, and better find out than risk it be a new infestation of an invasive. So thank you for paying special attention to the water weeds. Again, looking out for 2, Brazilian and hydrilla, and we're going to have a ton of our American Elodea in Lake Champlain. You will see it a lot, and you can practice counting your leaves on the whirl. I think you have these cards in your kits that are shared with you. Okay, thumbs up. So you'll have that side-by-side comparison. If you find something, you can sort of start to pick it apart and figure out if you think it's suspicious or not. Great. Okay. Carolina fanwort, Kabumba, Carolina. We intercepted this on a watercraft coming from Massachusetts last season. I was so glad it didn't get into the lake. All right, let's go to the next slide. So Kabumba, Carolina does look like a few of our native species, but it has one defining characteristic that's telltale. So it's a submersed perennial plant. It's not found in Lake Champlain. It is in states that surround Lake Champlain, so New York, New Hampshire, Massachusetts. Um, let's go to the next slide. I want to show you this defining feature. So the first thing to know about Kabumba Carolina is that the leaves grow opposite each other off the stem, but they have a segment of the leaf. It's like the leg of the fan. So there's like a stem, and then the leaf branches out. So our native plants don't have that stem that comes off of the plant. So opposite with the stem and then the fan is Kabumba Carolina, and the one that we're concerned about. We do have a native water buttercup and a water marigold, but their leaves grow without that section before the fan off of the stem. It's like they're attached right to the stem with the water marigold, and there's no, again, there's no length or piece there before the fan comes out. So water buttercup, there's some places in Lake Champlain you'll see that a lot. Water marigold took off last year. I saw more water marigold in Lake Champlain than I have seen in the last 17 years. So something's changing, and it's very happy. So I would expect to see a lot of Biden's Beckii and a bunch of the ranunculus species, and they may trip you up a little bit. And again, better to be forced to ask that question and see if it's Kabumba Carolina or not. So it's tricky, but again, we'll have plant keys and things for you to look at to be your own investigator and figure it out. Okay, and parrot feather is one of the only Myriophyllum species that is this, it looks like a plastic aquarium plant. It is very brittle and robust. So the aquaticum, I've only seen it once. It was growing in a water pond outside like a church property in southern Vermont. We got a phone call. It was planted, misnamed. We took it out and got rid of it and never seen it again. Never seen it in Lake Champlain, but let's go look at the species here. So again, a rooted submersed plant like the other Eurasian water milfoils, not found in Lake Champlain or in Vermont. It is growing in surrounding states and limited numbers of water bodies. It's not prevalent. Let's go to the next slide. So the leaflets on this plant are thicker. They're not as feathery. You'll have leaves coming off the stem in whorls of 4 to 6. So that means 4 individual leaves or up to 6 individual leaves coming off of one point on the stem. And there are times when it's flowering where you'll see these white flowers right in the axils of where the leaf comes into the stem, and it looks like white, like pollen almost. But that's the flower. So it's not always present, but that's a telltale sign if you can see it. Let's go to the next slide. Oh, that's it for parrot feather. You're not going to see parrot feather, but if you think you see parrot feather, please report it. Round goby, I spend a lot of my time preventing getting into Lake Champlain. This is a small benthic fish. I can't imagine a scenario where you throw a rake and you're able to pull one up, but you need to be aware of this species because there are a lot of anglers and visitors to Lake Champlain that think that we already have round goby, when in fact it's our native slimy sculpin species that they're talking about. Round goby is native to Europe. It first was introduced into the Great Lakes in 1990. It quickly spread throughout the Great Lakes and then moved across the Erie Canal to the confluence of the Mohawk and the Hudson River, where we're trying to prevent it swimming north up the Champlain Canal into Lake Champlain. Equally, they're in the St. Lawrence River and they're coming up the Richelieu River towards Lake Champlain. So the sign that you're looking for on the goby, so colors can vary, sizes can vary. The ones that I've seen near Lake Champlain are small, so they're only between 1 inch to 3 inches in size. And the round goby has a fused pelvic fin. So on its belly, it has a fused, it looks like a suction cup, that pelvic fin. So it's fused all the way around. Whereas our native slimy sculpin, its pelvic fin is splayed. It's not attached. So it's splayed open. And I just had an opportunity to go out on the Melosira, the UVM research vessel, and they were doing trawls and pulling up fish from the bottom of the lake bed to evaluate how our forage fish base is doing in Lake Champlain. And in and amongst all the little fish were a number of slimy sculpin, and I fell in love. They're very sweet and cute, but they're almost prehistoric looking. And round goby, being invasive, they tend to be a little bit hardier. So they like to live in shallow, rocky areas, in rivers and lakes, and they're very aggressive egg eaters. So they will eat the eggs of sturgeon, lake trout, salmon. They'll outcompete our native darters and our slimy sculpins. We're concerned about them because they can carry that viral hemorrhagic septicemia pathogen that I talked about earlier. And we are monitoring the infestations that are closer to Lake Champlain to make sure that they're not carrying it. But they're also changing the experience of anglers where they have large populations. So people will throw in baited hooks off the shore, and all they'll get time after time after time after time is goby, because they're such aggressive bait stealers. And in the state of Ohio, just as a fun fact, they will really beat up the bass population. And the way that they do that is bass can eat goby and get fat, but round goby will come to the nests of bass or trout or sturgeon, pick your species, and they will just constantly try to eat the eggs to the point where they'll exhaust the fish guarding the nest. And in the early season in Ohio, they had to eliminate the bass fishing because in the time it took to hook a bass, bring it up, take your picture, and release the bass again, the goby had swarmed the nest and eaten all the eggs. So they were actually starting to crash the bass fishery, even though the big bass were getting big and fat. They weren't able to produce any little successful fry because the goby were eating them all. So we're really trying to keep this one out of Lake Champlain. They do eat zebra mussels. They will never eat enough zebra mussels to have any impact on the population that we have in Lake Champlain. So that is one thing that folks who like goby will say is, but they eat zebra mussels. So the story of goby is still unfolding, but they are not present in Lake Champlain yet. And if you talk to people who think they've found goby, we really want a sample so that we can evaluate it. So encourage people, if they see you, and they likely will, and ask what you're doing. They can also be volunteer citizen scientists, and the best way they can help us is by bringing us samples so we can see what they're seeing. Okay, and then the quagga mussel. So the quagga mussel I call the evil twin of the zebra mussel for a few reasons. The quagga mussels in the Great Lakes have outcompeted the zebra mussels. They're more aggressive. They grow in deeper water. They reproduce more frequently than zebra mussels. It's just like the eviler twin, if you will, of the zebra mussel. So the way that we tell it apart is zebra mussels. At the fuse point of their shells, they look more like a D shape, and you can almost stand them up on their side. They sort of have a flat edge of the shell where it comes together. The quagga mussels are more rounded, and you have a very hard time standing them up. They'll just roll over. So they can get bigger. So we look for Dreissena bugensis on the bottom of watercraft and trailers that are coming, particularly from the Great Lakes region. We want to take an extra look for those. But if you happen upon a rock or some other species, you'll often find zebra mussels growing on the stems of invasive plants in Lake Champlain. Just double check them and make sure it's not a quagga mussel. We don't want those in Lake Champlain. Okay, and starry stonewort is the trick in the hat here. It's actually a macroalgae. Netolopsis obtusa is a rare macroalgae. It's only been found in Lake Memphremagog and in Lake Derby. So we don't think we have it in Lake Champlain yet. It grows again very close to the lake bottom. It sends out these almost like clear runners that try and sort of attach it to the bottom, but it's an algae, and then it produces these white star-shaped bulbils, which is a reproductive structure. Um, it has been confirmed in a number of other water bodies across the Northeast, and folks are pretty worried about it in the Midwest in particular, where it's growing in denser populations. So let's go to the next slide. So this macroalgae, you can pick it up, but its structure, you see it does not look like your Asian water milfoil. We don't see leaflets. We see sort of nodes with branching growth. Um, this one's tricky to identify. The bulbil would be a dead giveaway if it was present. But again, you're looking at segments of nodes and growth. The whorls that can come off of the stems can vary in the number of branchlets. So this one's tricky, and I would say by sort of an evaluation or an elimination of identification of other species, you might find yourself wondering if you found starry stonewort. It can look like some of our other native chara species that, Eileen or I can help you review. I think we only had 1 or 2 samples last year that came from the CHAMP program. So yeah, again, this one would be coming off of the bottom of the lake, so probably in the muck with the mud and might be confused with some other native, more finely growing plants at the bottom of the lake. All right, and the last one is water soldier. Water soldier, it looks like a floating aloe plant, is the simplest way to say it. It is a very robust plant. The spikes will cut your hands. This is a pretty nasty plant. It is not very well rooted to the bottom, so it can sort of shift around on the water surface. So it can grow underwater, but also at the water surface. As you can see, it's native to Spain, Italy, Bulgaria, and Siberia. We don't have it in Vermont. There was one population we knew of in Quebec that they're aggressively managing, which is an indicator that it can overwinter. So that's a concern, but we don't know it to occur in the state of Vermont anywhere yet. So here we go. Yes, it's submerged in the winter, so it sinks to the bottom and it overwinters. And then in the summer, with the new growth, it becomes buoyant and comes up to the water surface. These look like serrated, sharp leaf edges. It does have a white flower, but you wouldn't always see that. And I think this is, again, a telltale. If you pulled it out of the lake on a rake, I mean, it would be like a massive plant growth. Um, I'm I don't want to say anything, but I'm like, I want to say you will never see this. I'm hoping you will never see this in Lake Champlain, but we want to keep our eyes open. Okay, so that's the review of our 8 species that we have in the lake and the 8 species that we're hoping you will not find in the lake. And now I'll hand it back to Eileen to go over all of the methods and procedures for collecting the information.
Yeah, thank you, Meg. Something I just want to note here is you don't need to have all of these memorized right now. We provide you with a lot of resources on AIS identification as well as just aquatic plant species identification in general. So when you go out and survey and you get this mat of plants that you may not necessarily know, you'll have materials with you to reinforce what you've just learned from Meg. And I just want to take a quick beat and uh, open the floor to questions on any of those 16 plants or 16 aquatic invasive species that we just reviewed. If anybody wants to drop a quick question into the chat, or if you want to turn on your microphone and ask just before I dive more into the survey method side of things, you're also welcome to save it to the end.
Eileen, thank you for saying what you just said, because, and Meg, thank you for all of the information that you just provided. And I have to say, for me, hearing it the second time, it made so much more sense. So I'm really glad that we're recording this training so that people might, like me, who don't necessarily have a lot of grounding in botany, can go back over it and say, okay, here are the distinguishing features. And also, for me, myself last year as a volunteer for the first time, it was really helpful to have the hands-on training and to be able to like look at some of the samples that you brought. So it looks like you do have a question in the
I see
chat.
the first question. Great. So, how would you find golden clam when you're throwing a rake from a boat? That's a good question. So, when you do a rake toss and you throw it from a boat, you sort of give it a moment to settle down to the bottom of the lake. And often at the lake bed interface, like where the sediment is, when you hook up some plants, you'll often hook up a bit of sediment or mud, and that is where the corbicula or that golden clam might be hanging out, and it could get caught. And in fact, that's what happened to the volunteer who was monitoring at the South Bay boat launch. So they threw their rake in the water, they got some plants, but they also got a little sort of a cup of mud that came with it, and they went through it, they sorted through it, and they found half of a shell of a golden clam and said, oh, that doesn't look right. So that's probably how you would encounter it if you were throwing a rake from your boat.
Okay, thank you. Any other questions? I'll take also just at the end of the presentation. But I'll just start off with how you conduct a survey for CHAMP from start to back, starting with the materials you'll take out with you. So you'll want to bring aquatic invasive species rake, data sheets. You want to bring your phone so you can take pictures and you can get GPS points. If you have other devices for those two purposes, have at it. I think a phone is an easy single thing to bring with you, but please feel free to bring a camera and a GPS device as well. We also provide you with a jeweler's loop, which is a little magnifying glass. We give you plastic freezer Ziploc bags for specimen collection, and we give you a white bin, which is good for viewing specimens in a contained area, and it's very helpful for photographing your specimens. Some optional materials. There's two survey methods that I'll go over. You can survey from shore or you could survey from a boat. So it's optional for you to bring the boat and everything you need with it. And we require that you wear personal flotation devices if you do survey from a boat. You can also bring a cooler with you for specimen collection, and you should bring sun protective measures. And just something I want to note here, we really love pictures. So have that phone or camera handy and take pictures to accompany the survey. So this is just the top of the data sheet that you'll go through for doing your chance survey. You fill in your name, your phone number, your email address, the name of your site, which survey method you're doing. So if it's surveying from the shoreline or if you're surveying from a boat, I'll go over start and end points and start and end time. But you want to characterize the relative water level, and that's relative to the time of year at your site, what you would normally be seeing for the water level. So you characterize the water clarity. We don't give you any actual quantitative devices for measuring water clarity. We just want you to do your best. It's a subjective measure, but say if it's relatively clear or if it's highly turbid, you would circle poor. You characterize the light conditions of the day. So if it's sunny, not a cloud in the sky, or if it's completely overcast or somewhere in between. And then you circle if the surface conditions of the water are calm, which is relatively flat, rolling, or if it's rolling so much that you can see the tops of waves forming a white cap. And then you describe your survey area, which I'll go over in a second. Your survey area can be a maximum of a quarter mile long, but it can be as small as just a boat launch area with a dock. What's important is you capture that survey area and its start and end points in your survey description and in the start and end point GPS points. So those start and end points are the ends of the survey area which you survey between. Each survey consists of 10 rig tosses spread out evenly throughout your site. When you survey from shore, you want to space those out linearly along as you go. And if you survey from boat, you want to paddle out a little bit further into the water and sort of snake your way around throughout the survey area. And you go out as far as the littoral zone, which is the zone in which light reaches down far enough into the water for substantial plant activity. And this varies by site, how far out that can go. So for the section of the data sheet that asks you to describe your survey area, you just want to use landmarks when you can. Permanent standing structures are really helpful just for when we're sort of ground truthing where your survey is actually taking place. If you're at a boat launch, you might describe the dock or the docks that you survey from. Or if you don't have docks, describe the area of survey that you go between. So an example of this is rake tosses moving perpendicularly from shore along a north dock, and then moving south along the shore between the docks. Final rake tosses moving perpendicular from shore along the south dock. That's if a site has two different docks. So to conduct a CHAMP survey, and this goes for both shoreline surveys and for surveying from a boat, prior to your first rake toss, you want to get the time at which you start the survey and the GPS point, and you record both of those on your data sheet. You can get your GPS point by using a navigation app. So you can use Apple Maps or Google Maps, or if you use a different app, I'm happy to help show folks how to do that. And if you don't bring a phone with you, I will also work with you to get those precise start and end points. So you get your time, you get your GPS point of that first toss. You then do another 8 rake tosses spread out as evenly as possible throughout the survey area. And you record those findings cumulatively onto the data sheet. As you go along, you want to not just be going based off of what you find in the rake toss. You want to also be looking at the water. This is especially important for shoreline surveys because it takes a little bit more effort to lean over and actually check the water as you go. And then you record the aquatic invasive species that you find and photograph them in the white bin that we provide you with as you go. So all invasives that you find in your survey, please support with a photograph. So that's 9 rake tosses. And then at the 10th and final rake toss, you should be at the endpoint of your survey area where you will then record your second GPS point and then the end time as you get that final toss in. So for folks surveying from shore, what you want to do, so the AIS rakes are 2 raked heads zip tied together so they both face out with tines facing away like that, tied to a length of 25-foot rope. So you want to take the end of the rope away from the rake heads into your non-dominant hand, sort of make sure everything's untangled in the rope, and then take the rake head and stand at the water's edge and throw the rake into the water through to the whole length of that rope, or as far as you can within that length. Give the rake a moment to settle to the lake bed, because that's where you're getting a lot of those submerged species, and then slowly pull it towards you. As you pull it towards you, it'll collect, depending on your site, either a few plants or a massive mat of plants and mussels and biomass in general. So you want to take this mass and use the white bucket to sort of just pick through it very thoroughly. And you can use that the bin to help you view species in isolation. and examine and then record onto your sheet what you find, if any of them are invasive species. It is very important to be thorough here because, as Meg mentioned, when we found golden clam last year, it was because somebody picked through a huge mat of biomass to find one teeny little golden clam. So you really want to take your time digging through it. The clams and mussels can attach themselves to plants, to sediment. Just be as thorough as you can. And then for rake tosses from a boat, it's sort of the same idea. First of all, you're required to wear personal flotation devices, so that's life vests. You do your 10 rake tosses interspersed throughout the survey area, which is the littoral zone. If you're surveying from a kayak or a canoe, it's important to note that it's very common that as you pull in a rake toss, it'll pull your boat towards your rake rather than your rake towards you. That's okay as long as you're getting a chunk of biomass onto the rake as you pull it up. That's still good data that you're collecting. And again, you use the white bin provided from your Champ toolkit to examine what you collect and be thorough. Something to also note for folks surveying from a boat that just between having all the toolkit materials with you, as well as all your paddles and materials you need for operating your watercraft, it is a lot better to go with a buddy for surveying from a boat. And we also have some volunteers who will collect their rake tosses as they go and then do a species inventory on shore. And if you do this, just make sure you know as you go the density of the aquatic invasive species as you collect from your rake tosses. I'll talk about that parameter in a minute as I go through the survey more. So this is what that species inventory looks like. So here we have the target species all listed out. You just give it a check if you find it. Then you characterize its distribution as either trace, sparse, moderate, or dense. And then any relevant notes about perhaps the size of the bed that you find. If you see zebra mussel, maybe if it's biofouling something at a dock, or just any other kind of notes that you want to include. And it's a similar thing for all the watch list species. Something I want to note about what aquatic invasive species distribution means here. This is a parameter that looks at what is captured in your rake toss rather than what is observed in the water. So when you find a trace amount, you're looking at 1 to 2 fragments captured into your rake toss, a fingerful. So it's one or two tines of the rake have the plant or the species on it. Sparse is 3 to 6 tines. Moderate is the majority of the tines, and dense can be almost all of it or a large mass covering the entire rake. If you do 10 rake tosses and you find one dense and the rest are none, just note that in the notes column. So for sample collection, this is not going to be done with necessarily every survey that you do. This is just the protocol that we want you to follow if you find something that you believe is on the watch list of our survey. So those 8 species that are not yet in Lake Champlain. You can also do it if you're just a little bit suspicious that it is a watch list species, or if you want a confirmation on the identification of that species beyond what you can capture in a photograph. So what we ask you to do is photograph your specimen and fill in all the details about it on the survey data sheet. And then you will, using the plastic bags that we provide you, you collect and store a sample of as much of an entire plant or mussel as you can get. And then on that plastic bag, wrap it in a wet paper towel. And on that plastic bag, write your name, the date, the name of your site, and any notes you want to include. Keep the bag in a cool place, so refrigerate it, until you can send the sample to either the Vermont Department of Environmental Conservation or the New York chapter of the Nature Conservancy. There's full page instructions on how to do this in the manual as far as the mailing portion, and there's also mailing labels that you can cut out and include in an envelope, and you just wrap up your moist sample, seal it, put it in an envelope, glue or tape the mailing label to it and send it off. So that's for if you see a suspicious species. Something that is new this season is that we're also asking folks to conduct a quality assurance sample as a part of their surveys. Just one survey out of the season, so one of the three, we're asking you to collect and submit a quality assurance sample for us and for the Lake Champlain Basin program to review. So I suggest doing this for your first survey, not just to get it out of the way, but also if we're running into issues with species identification, it's good to catch it early, and then we can work through what is and is not being identified in a sample. So you start by labeling your bag with your name, date, site, and QA sample. This just indicates to us that this is your quality assurance sample. You gather a gallon plastic size chunk of biomass into that bag and seal it off. And you want to make sure that this biomass contains the aquatic invasive species that you reported in your survey. So I would suggest after your first few rake tosses of your first survey, say you find a few Eurasian water milfoil and you find a few zebra mussels, you want to then have a representative rake toss, one that would have a few of these Eurasian water milfoil and zebra mussels, and put all that biomass, not just the milfoil and the mussels, but everything you're gathering from that rake toss into the labeled gallon bag. And then for the drop off of those, you can coordinate with me. You can either drop it off in LCC's office Monday through Friday. You could drop, or you could also mail it to us. You could also drop it off with the Lake Champlain Basin Program boat launch steward. I can share out where they are located. They're spread out throughout Lake Champlain, so it might be easier to get to one of them than it is to get to our office. And if those don't work, we can coordinate pickup where I come to you or we meet at a midpoint to pick up your sample. So to submit your CHAMP survey, once you have everything filled in, you wanted to transcribe your data from your field sheet onto LCC's online form. We'll provide you with links to these forms, and also on our data sheets, we have a scannable QR code so you could submit your data from your phone. And you just this has parity to what you're surveying for on those survey data sheets. So you just copy it over exactly as how you have it written. And please add photos. And if for some reason they're too large to attach to the survey, which happens, or if there's just too many to include, please feel free to email me and or Lindsay with the pictures of what you're surveying for or what you report in the survey. As you go through, you want to just go back and check every section for accuracy, compare it to what you have written, and then you hit submit at the very bottom of the form, submit CHAMP survey. And after you hit submit, it'll prompt you with, are these values correct? You get to do one more check, and then to hit send, you hit next here at the bottom of the page. So that is how you do a CHAMP survey. Just to go over the next steps now that you are trained in the background of aquatic invasive species in Lake Champlain and how to do the surveys. I'll be reaching out via email next week with a digital copy of the CHAMP manual, as well as links to submit data, additional resources, and a recording of this training so you can go back and watch this again. And I'll also have information about the upcoming in-person trainings, which you should have signed up for on the Champ volunteer sign up form. Um, Between now and those in-person trainings, I ask that you think about where you want to do these surveys, where you want your sites to be. Um, and I'll confirm that with you at the in-person trainings between June 6th and June 19th. Um, and if you need help determining a site or if you want one assigned to you, or if you have any other more general questions, please reach out to me and to Lindsay. So now I'll open it up to questions. I think I saw one pop up in the chat as I was going. Let's see here. So Paul asks, how should we match photos with data point? It's a good question. So do you mean with the data point as in the point of the rake toss?
I mean, the location of the data point being we do 10 different points and we record all the GPS and so on. But when you take a photo, although I did just find one place on an Apple photo where I can theoretically change the location information to say it was data point number 1 or data point number 7 or whatever. Do you have any other ideas or plans?
We're not asking folks to match the photos to a data point. It will just correspond with the survey overall.
Okay.
If you would like to do that with matching it, that's fine, but it's not part of the CHAMP protocol. It'll just match with the survey. Beth asks, do we use the same start and endpoint each time /survey? You can, but you should mark it each time just in case you're standing a little bit off from where you start and end in your survey. But for at your site, your start and end point would be about the same for what you're using for the same site name. Does that make sense? Sounds good, Pasquale.
So Eileen, I have a question.
Yeah.
Oh, sorry, Pasquale, I didn't mean to cut you off. Oh, okay, that was just an intro. Sorry. Um, so the photo requirement, I think, is new this
Mhm.
year, and it sounds like if we have a suspicious sample, we want to have the actual sample and also a photo,
Mhm.
but also anytime on the regular survey we encounter an invasive species, we want to take at least one photo of that species and submit it. Is that right?
Yes, that's correct. So if you're finding on every rake toss you're using water milfoil, you only need to photograph it once. We do welcome more than that, though. Photos are awesome. They really are. It just helps us sort of have that double check on things.
Well, I think, Meg, thank you for sharing the story about exactly how the golden clam was discovered because I didn't know that prior. And I think it really illustrates the importance of the back and forth. And I know I only did one survey last year, but it was definitely a learning process just over the course of it. So I really encourage people to reach out with questions and to be very thorough. And I would say also, Eileen, you mentioned that if you're in a boat, it's helpful to have a partner. I think it's helpful to do the survey with someone else, even if you're not in a boat. And I did my survey from the shore, but I recruited somebody to help me. And it's just nice to have another set of eyes actually there. So I'd encourage people to do that as well.
Definitely. Okay, any other questions? Well, amazing timing. Look at that. Um, Jenny, you could stop the recording now.
Yes, I will try. I almost accidentally ended the meeting, which is not what I meant to do.
Don't worry about it if it's yeah, I'll just edit it out in the